It's a pleasure to be here, and it's a great time for neuroscience in Europe, I think, to have this event here. And I feel privileged to be here today talking about a project on autism. So we'll talk about EU AIMS, and that stands for European Autism Interventions, a multi-center study for developing new medicines, new medications, EU AIMS. And this is a presentation on behalf of the consortium, and my fellow leader on this project is Declan Murphy, which is an absolute pleasure to work with him. So what is autism? So autism is characterized by a set of symptoms. Core symptoms include social communication deficits, repetitive behavior, and language disability, and a set of what is called comorbid symptoms that include sleep deficits, mood, anxiety, attention, hyperactivity, a cluster of symptoms called irritability and seizures, and like most other CNS disorder, it comes also with non-related neuroscience effects such as GI disorders and immune dysfunction. The incidence of this disorder has increased massively, where it was just a few years ago a rare disorder. It is now more, the incidence is higher than, for example, schizophrenia. The current indication is now 1 in 88 births. It's 4 times more frequent in males than it is in females, and it has a very strong genetic link, maybe up to 40%. Some people claim that it may be as high as 50% or even higher. There's no treatment for core symptoms, and there are 2 antipsychotics indicated for the treatment of irritability, which is Risperdal and Abilify, and we currently believe that autism is a synapse disorder. So there are no treatment for core symptoms, but there is medication used in this order. There are anticonvulsants, sleep medications, antidepressants, and stimulants. And what I would like to say about these treatments is that they're mostly non-evidence-based, meaning that they're anecdotal evidence. It's trial and error. Most of it is, or if not all, it's off-label use, and most of them don't have a particular pediatric label to treat children. So it's a typically bleak place to be in. So what's the situation for autism spectrum disorders in 2012, just before we started this project? Well, there's no major strategy defined in Europe. There's no major or concerted effort in drug discovery. There's no preclinical network. There's no translational network. There's no regulatory strategy. In general, there's late diagnosis and poor awareness, particularly in adults. There's poor knowledge of patient needs across life course, particularly in the transition from teens into adulthood. And what I already said was that there's a wide range of treatment strategy with no evidence of efficacy. It's trial and error. So, imagine yourself being a parent of a 2-year-old little boy that has just been diagnosed with autism. Where do you go? And then there's silence, indeed. It's a pretty awful state to be in. So if we want to move this field into evidence-based treatments, it's very clear from this slide, I think, that this can only be done in a concerted effort, such as an IMI project. There's not a single organization who can tackle this, not a patient advocacy group, not a pharmaceutical company. It has to be a concerted effort. Now, why do we believe that the time is right to do this and set up this major journey for us and develop neurodevelopmental disorders and put them on the European agenda? Well, there has been a small revolution in neurodevelopmental disorders as we moved the field from being a sin, basically being bad parenting, particularly mothers were blamed for that, to a field that is called synapses. So basically what we understand now that neurodevelopmental disorders are a disease of the synapse, basically developmental maturation, stabilization of synapses that is not well regulated. And we believe there are 2 extremes in this that is indicated in the graph on the left bottom. In the middle you see a normal neuron which had these little sticks, which is spines, and on these sticks you have the synapses, which is glutamatergic synapses. That's a normal situation, and the two extremes are, we believe at this stage, is Fragile X, where you either have too many synapses that are immature, or on the right-hand side, too few synapses, such as in Rett syndrome. That are the two extremes, and we currently think, and that's a hypothesis, it's a gross generalization, of course, that most of the ASDs that we know will fit in this spectrum. We have identified about 350 genes involved, and they somehow all have some role on the integrity of the synapse. So that's where we are today. And on the right-hand side is a cartoon that sort of hits at the key stage where we are at this moment. So we have established the genetics, and we have established molecular pathophysiology and target ID. So basically what in a pharmaceutical company you would say that we're at the target selection stage. Now what we need to develop is translational model. This includes cellular models and animal models, mechanistic therapeutics, and clinical outcomes and diagnostic biomarkers. How can we do this? Hopefully EU AIMS will help to do that and helps to create value for these patients. So EU AIMS is an unprecedented public-private consortium in this field. We have 13 or 14 academic partners, small SME, a few SMEs. We have 6 pharmaceutical companies on board, and we're very happy with the presence of the leading patient organization in the world, which is Autism Speaks, located in the US, US-based, and they're also actively sponsoring us with $1 million, so this is very favorable. EU sponsors us with around 30 million euros. On the right-hand side is the strategy that we're taking. We're taking a relatively simple strategy in terms of how we want to move this to new evidence-based treatments. So on the left-hand side, you have cellular assay work stream, you have a work stream on animal models, you have a work stream on translational sciences, a work stream on clinical research, and then everything is tied together by a work stream on bioresource and data management. And this consortium was launched in April 2012. So anything I'll present next, you need to see in the context of the activities that we have since our launch, which is April last year. So now I will highlight some of the successes that we had. So we had 12 publications in the first year of the project, and these include top-line journals. Now I know that impact factor doesn't say too much. That was mentioned this morning earlier, but I think it does tell you something about the quality of the work that was done. So we published in Nature Drug Discovery, Science, PNAS, Molecular Psychiatry, JAMA, TIPS, and a couple of other papers. And the mean impact factor that we have over these papers is 30.8, which is pretty high, I would say. Neuroscience Roche, or Hoffmann-La Roche, is aiming for a mean impact factor of 7 for this year. So we're twice that, so it's a very good place to be in. Next to that, we were on the cover of 2 journals. We were on the cover of Nature, and we were on the cover of Trends in Pharmacological Sciences. So now we'll highlight some other achievements. So one of the papers that we were on the cover was about the father figures. So if you see the graph on the right-hand side, which is basically looking at a correlation between the number of de novo mutations and with the age of the father at the conception of a child. So you see the function of the number of mutation of the age of the father, and what you see is that there's a linear correlation between that and the risk of having a child with autistic or schizophrenia. That's very interesting information because, as we know, we tend to get, families start a real family much later in life, mostly now in their 30s, and there's a clear risk associated with that, as you can see. And this study then highlighted that the age of the father is a risk factor for a child to have a mutation leading to autism. Another achievement that we have is that we identified a new target for the treatment of autism. And this was a study run by the University of Basel, a local study together with Roche, and that was published in Science. And what we have done here was used the Neuroligin 3 knockout model mouse, which is a model for autism and has construct validity. The mouse has deficits in the core, what we call core domains, so social behavior and stereotypy. And we looked at the changes in neurochemistry and neuropharmacology, and what we found there was a selective upregulation of mGlu1 receptors. So this is very interesting. This was seen in various areas of the brain, that's very interesting because it's also probably a druggable target. We're actually in the process now of translating this into the clinic. We're looking at autoradiography in these mice and also in human tissue. We're currently discussing with Merck, who has a PET ligand for mGlu1, as we want to follow this up into the clinic. At the same time, we discovered that mGlu1 might be a target for the treatment of autism. We did something else. We basically said also that there's a shared synaptic pathophysiology in syndromic and non-syndromic rodent models of autism. There's a controversy whether comorbid phenotype is really a similar form of autism. What we actually show now, and that's been very well established, that Fragile X, which is a syndromic form of autism, is, by the work of Mark Baer, is linked to over expression of mGlu5, which is another mGlu receptor. And we're saying now that with this study that there's convergence of pathways, mainly on the glutamatergic way, within the group 1 of the mGlu receptors. So there's shared synaptic pathophysiology. Then we have major achievements on the regulatory field through the wonderful work of Maria Isaac, for which we are very happy with. Because we've been able to do this in the first year. So what we've been able to do is to move European Medicines Agency to draft a concept paper on the development of medicinal products for the treatment of autism spectrum disorder. And this regulatory guidance document was initiated in March, and this concept paper will be expected in 2014, which I like to add is a major success for this field. We finally have it on the EMA agenda. And as you well know, there will be no drug on the market without the regulatory approval of EMA. Now here's some other headline achievements. So we have developed Rebos methods to reprogram keratinocytes into IPS lines from autism patients, and these lines have now been distributed across the EU AIMS partners. Within Work Package 2, on the animal models field, we have set up a central animal repository that was established in Harlan now in Italy. All transgenic lines will be moved there, and all the partners of EU AIMS can order them from there. We have set up, completed, and have the results now of a multi-site study for standardization and cross-site comparison across Europe. There were 4 sites involved testing oxytocin. Oxytocin is a peptide that is thought to be critical for treating social behavior. It's a gold standard, we think. And we have now the first results, and there seems to be an excellent correlation between sites across Europe. That's very good news, because standardization and having results from different sites is not always an easy case. We have genetically rescued the Neuroligin 3 knockout mice. We identified the target, and we're now moving forward with pharmacological intervention studies to see if we can improve the behavior of these animals with mGlu1 receptor antagonists. And as I said, we are moving this forward into man, translation into man, with a possible PET study. Also very happy to report that we strike an agreement with Sage Labs, as I think we're moving from transgenic mouse into transgenic rats. And we achieved a deal where we can have a 50% reduction on transgenic autism rats. So this is a new area that we're moving into. A mouse is not a small rat. We can do much more behavioral analysis with the rat, and the brain is also bigger. That helps as well. So the first transgenic rats have arrived at sites across EU AIMS, and the testing has been initiated. And then we had also market success in translational science and clinical sciences. So we actually have evidence, the first human evidence that brain functional abnormalities can be reversed in adult ASD by modulating 5-HT. That's currently being tested as an outcome predictor for clinical trials. And we have new data which are published, I think, that shows that ASD patients have significant differences in GABA alpha 5 binding. That's also good news because there are drugs around that selectively bind and modulate subunits of the GABA channel. On clinical sciences, we have new data sets as well. We've been able to show that there are differences in cortical anatomical connectivity. This is now being tested as an outcome predictor for clinical trials. We have new data on risk factors for autism that have significant differences in brain functional response to emotional sound. This is now also being followed as a risk predictor. What we have initiated in the beginning was a survey of how we actually treat autism patients in Europe. We don't know. There's no record of our prescription behavior of autism patients across Europe. So there's absolutely no sense of what these patients receive in Europe. So we published now a first study. We did a survey, will be published soon. And what we basically demonstrate is that there's very low prescription rate for associated symptoms, and there's very wide, with emphasis on very, variation across different countries. And one of the highest prescription area is the Netherlands. It's the land of my origin, which surprises me, but that's what it is. We obviously had regulatory success. We worked with EMA to launch concept paper for regulatory guidance. And we set up a biorepository and database, and that will soon be receiving lots of samples because we're moving into the clinical arena. I think these are the major headline achievements. I think we've done a lot in the first year. It could always been better, but I think we have an excellent data sheet here to present. So what's next? Well, we move into our clinical studies. So we used the past year to set up studies in patients. One of the things that we're going to do is to look into high-risk sibling study to investigate patterns of brain development that may be associated with early detection of ASD. As you may know, the general age of a diagnosis of a child with autism is around 3.5 years or even later. So the earlier the better, and if there are drugs on the market to treat them, they may also be, they may have a better efficacy when they're treated as early as possible. In this study, we'll use infants between 3 and 7 months. We're going to look at substantial numbers, so 300 high-risk infants and 100 low-risk infants. We will do MRI and NRS, which will allow us to look in deeper parts of the brain, particularly looking at some blood flow changes, some functional data, and white matter development, infant brain processing of human voices, looking at more at the emotional side, so it's going to be functional data as well. The other very important study, as far as I'm concerned, is the accelerated longitudinal study. This is a observational, naturalistic study in autism patients with huge numbers, so 400 ASD patients and 300 healthy controls. And we're just going to look how we're going to recruit them and then just follow them over the course of time, because we do not know what the medical needs are of these patients over time. For sure, the medical needs of a 6-year-old is different from that of the medical needs of a 14-year-old or even a 30-year-old. And this is something we're going to look at. We're going to map that on the agenda. We're going to recruit patients, young and old, and we're going to follow them for 2 to 3 years, just to increase the knowledge of patient needs across the life course. It may seem odd to you, but that has never been done before. This is an unprecedented study. We're building, as we speak, a clinical trial network. We've contacted various partners from European networks. We've sent out in the first year 48 invitation letters to 41 sites. We got a huge response rate. There are 34 sites from 15 countries that show willingness to participate in the clinical trial network. That is 83% of the sites contacted. The little flags that you see on the map right is the distribution of these sites. Please note that the flag may indicate multiple sites in the same city. What you also see is that the Eastern European countries look a little bit bleak, as well as the Scandinavian countries, so we will send out a second wave of letters focused on Eastern Europe and Scandinavia. Furthermore, I'm very happy to report that EU AIMS has reached a stage where we no longer have to contact people, but we get contacted. So in the past couple of weeks, we have been contacted several times from sites across Europe, but also from Israel, for example, who wants to join our clinical trial network. And we have also been contacted by a Chinese consortium on autism to see if there could be an exchange of information. Last but not least, we want to develop a European inventory of ASD patients and measures that will help us with data compatibility, and we want to coordinate large clinical databases that probably will steer us towards identifying potential outcome measures. What are the remaining challenges? Well, the list could be endless here. I think one of the key issues is the heterogeneity in patients, so looking for relevant biomarkers that predict efficacy of the drugs that we want to develop. Ultimately, the evidence-based therapy is an issue. Let's be very clear, if we start now a project, it will be 10 to 15 years before it's on the market, so evidence-based therapy is far away. I think we have, in general, a concern around the clinical endpoints. We are thinking about the sensitivity of these clinical endpoints and the specificity of these endpoints. And in general, I think the general awareness of ASD across Europe is appalling. So it's very poor. So I think we have a huge job here. What I like to stress is that if you want to develop a drug in schizophrenia, in depression, in Alzheimer, there's a road already ahead of you. And we are actually paving the road while we're driving the car. So we're paving the road just in front of us. So we're paving the way and we can make the next step. So that's where we are. So it's very challenging, but it's a pleasure to do. So this is the consortium. We're a wonderful consortium. We are really working very nice together. You can find full details of our program in this paper, which we managed to publish in Nature Drug Discovery last year in November. If you have still questions, feel free to contact us. This is my email address and Declan's email address, or the project management office. You can find us on the web, euaims.eu, and you can find us and you can follow us on LinkedIn, where we regularly report progress. Thank you very much.
Thank you, Will. Questions, comments? I would like to ask you a question then. I don't know if the question is for you or for Maria. Obviously, these projects, and the reason why industry as such is interested in this, is because we see this as connecting, you could say, the patient with basic science. And obviously, one of the important steps is to get the work from basic science recognized by the regulators. And my question goes, now we see EMA has picked this up. What are you doing, either Maria or Will, to also get FDA, PMDA, SFDA, the list is endless, to pick this up so we can secure that patients not only in Europe will benefit from this?
That's a question for Maria.
No, thank you so much. I agree with you, because here is a lot of people that is very interesting. We work very close with FDA, definitely in relation with clinical endpoints, trying to have common understanding in this kind of diseases. And like I say, it's usually, I think the difference with this project was that from the beginning, we only have one opportunity to do something like this. People like us, we see it. It's been a hard work. Suspicious at the beginning, I think it will, and I can understand. But I think it is, as long as you work for the patients and you believe what you are doing, FDA is very interesting. We communicate, let them know what is going on. And the guidance in the beginning is only for symptomatic probably, but the importance is that it's the first time, I agree with him, that in regulatory view it was a lot of complaints, suspicious, are you treating, really needs treatment in Europe? And in Europe we've been very tough with ENS. This is a disclaimer, just in case I don't have a job when I come back, but it's true that I agree with you, it's been very difficult.
Yeah, okay, thank you. Yes, please.
So you said that for evidence-based medicine, we may have to wait for another 15 years. But there are many, as you said, off-label drugs that are prescribed. So why not look at those and try to get some evidence behind them? Because that may take less than 15 years, I would hope.
Yeah. Well, it's absolutely a good point. So that's it's not within the program of EU AIMS, but it's certainly a way forward, I think. It would be good to have biomarkers that sort of will make sure that the right patient gets treated. So we need some kind of clinical study for a specific product, but be it an antipsychotic or an antidepressant, where in a subset of patients this may work. And this needs to be done, so I totally agree with you.
Okay, thanks a lot. Will?